Okay, so welcome to the lecture on DNA replication. This one does not have a PowerPoint. This is strictly done by illustrations on the whiteboard up here. So we have here a segment of DNA. This is a particular snippet of a much longer DNA strand, a chromosome. We're going to show a step-by-step instruction, so to speak, of how DNA is replicated. So a couple of things. First of all, note that this is DNA. It is double-stranded. The nitrogenous bases thymine, adenine, guanine, and cytosine are represented by A, C, T, and G. Remember, specific base pairing, A always bonds with T and C always bonds with G. Keep in mind there is a hydrogen bond between each of the members of a base pair. So that's part of what keeps DNA wound together as that double-stranded molecule. So we have a single strand of DNA. We need to duplicate it. DNA is said to be what's called a semi-conservative replicating molecule. Here's what that means. What that means is that DNA starts off with the two strands, right? They're split apart from each other, and half of each is produced by new nitrogenous bases coming in and synthesizing that new strand. And so each new strand conserves half of what we started with. Semi means half, semi-conservative. And so if you can visualize that happening to this whole thing, it's going to split, and we're going to add half, or one new strand, to each of the original strands that we started with. In order for DNA replication to occur, several enzymes are going to be necessary. If you remember, an enzyme is a protein that speeds up a chemical reaction. In this case, a number of reactions are going to occur, a number of things are going to happen, and enzymes are needed for them to happen. I'm going to use very simplified shapes to represent each of the enzymes, so please don't think this is what they actually look like. It's just easier for me to use the more simple shapes. So without further ado, let's get started. The very first thing we have to do is break apart all of the bonds between the base pairs. Because if we're going to separate the original strands from each other, we have to separate those bonds. And so an enzyme comes in to facilitate the breaking of these bonds between members of a base pair. The name of this enzyme is helicase. And so helicase is going to separate those strands. You might want to fast forward through the next minute or so, because I just have to show that happening up here. The good news about specific base pairing is that I can look at the top half here and know exactly what letters I need to use. So helicase has unzipped the DNA separated the two strands, and it is continuing to move like we see here, unzipping the DNA as it goes along. Now, those bonds between members of a base pair, they're strong bonds. And so what we need to do is we need to have something that's going to prevent them from simply reforming those bonds due to attraction between members of a base pair. And the molecule for that task is going to be the single strand binding proteins, often shortened to SSBs. So just a reminder real quick, this is helicase, and tell you what, I think it'll be better if we have a little key up here. So this is helicase. One of these days I'll get a black marker that actually works well. This is helicase. These orange molecules are the single strand binding proteins, typically shortened to SSBs. So they keep the strands from reforming the bonds together, which is great. That's what we need. Now we need to start synthesizing the new complementary strand. However, the primary workhorse in this, which is going to be DNA polymerase, it can't start from nothing. It needs a springboard. And right now there's no springboard. So we need an enzyme that can start from nothing to get this thing going. And that enzyme is going to be RNA primase. Prime means first or foremost, and so RNA primase is the first and foremost enzyme when it comes to laying down the complementary strand. So RNA primase comes in. Let's see if I can get a brighter red for you. There we go. RNA primase comes in and lays down the first three letters complementary to the original strand. Since it's RNA, it's going to have a U instead of a T. And it's going to do that, and then it's going to leave. Now we have a springboard. We have a starting point. So, what comes after RNA primase is the true workhorse in this, which is DNA polymerase. Now that it's got a starting point, DNA polymerase can start laying down the new strand. And it's going to keep going, effectively playing follow the leader with helicase. As the helicase unzips, the DNA polymerase synthesizes the new strand. Seems pretty simple. Eventually something will have to come in and replace this little piece of RNA with DNA, but that's something that happens a little bit later. So we've got DNA polymerase following the helicase. All seems to be going pretty well. I bet your next thought is, why doesn't the bottom half just do the same thing? Well, there is a problem with that idea. And here's the problem. Let me see which side of the screen I have more space to write on. Looks like the right side. So, I've got two markers here, and I'm holding them for you parallel. However, if I move it like this, they're still parallel, but now they're anti-parallel. They're running in opposite directions, but they're parallel as they do it. DNA is like that in the sense that there are two ends to DNA. One end of a DNA strand is called the 5' end. The other end is called the 3' end. We're going to say the cap of the marker is going to be the 3. So if the top one is running from 5 to 3 like this, the bottom one has to be running from 5 to 3 the opposite direction. Okay, so DNA polymerase is great at what it does, but it can only work synthesize new DNA from 5 to 3. So that means, in our example over here, this new strand we're building is going to be the 5' end, working this direction towards what ultimately will become the 3' end. That means this top part has to be 3. And down here, this would be the 5. So this bottom part runs from 5 towards the 3. The top part goes from the 3 towards the 5. Polymerase is building then the new strand that's going to go from 5' to 3'. However, down here, if we want to build the direction of 5 to 3, we have to build backwards. And that's exactly what's going to happen. Primase comes in after helicase has opened up some space for us. Primase comes in, lays down the first 3 letters. A second polymerase comes in. And continues the job all the way back to the start. By the time it gets that back here, helicase will have opened up more room. The primase will then go, lay down a primer here. The polymerase will follow it and then start to build backwards until it juts up right about here, because it can't actually join together with the primer that's there. And that whole thing just continues to happen over and over again on this bottom strand. The top strand is referred to as the leading strand. It's called the leading strand because it tends to occur first. The key with the leading strand, it's a continuous build. Once helicase unzips, the primer is laid down, the polymerase just builds continuously without stopping. The bottom strand is referred to as the lagging strand, and it builds discontinuously, which means that the primer has to do its job more than once, laying down, primase has to do its job more than once, laying down a primer, and then backwards building from there. This space that occurs between chunks, so this space right here, that occurs between chunks of DNA built on the lagging strand, this has to be ultimately bridged by an enzyme called DNA ligase. It's going to bridge the gap between the fragments of constructed DNA on the lagging strand. And those fragments are called Okazaki fragments. This is one thing that's really, it's best viewed in an animated style. I've linked a video on YouTube on the Moodle page that shows this pretty well animated, so you can watch it happening as it goes. Keep an eye out for the enzymes in that video, and I think it will be really helpful for you to help visualize a bit more what's going on here.